This is a production of Cornell University.
So, today I would like to talk about the fact that peas can be used as a model plant, as they were many, many years ago before the miserable little weed interjected itself. But there are things you can do with peas that you can't do with Arabidopsis because of its corline or stem nature. But I would like today to talk about the modern findings in peas. And here's the subject. But the reason I got into this again was actually because of Professor Emeritus Chris Ween, that those of you who are long-term members in the department will know well. Well, Chris called me up a couple of years ago and he said, I'm revising my vegetable crop physiology book and I need somebody to write the chapter on peas. I said, no way, Chris, I'm retired, forget about it. So he went away. He then came back a year later, in other words, about a year ago, and said, Peter, the guy who was going to do it has failed to produce. I really need a chapter on peas. I said, okay. So I had to do a lot of research on peas in the literature to bring myself up to date. And even though I worked on peas, my graduate work, my postdoc work, and a lot of my career, I wasn't up to date. But as I spent months getting up to date, I thought I would share the knowledge I gained. So garden peas. We know them as garden peas, but they can also be crops. And most of you probably, if you don't grow them in your garden, you probably know them just from the supermarket freezing chest. But the peas are grown as one of the major crops, not up with the big corn and soybeans, but they're a major crop in many countries. So here's the global pea production, and we can divide peas into green peas and dry peas. So here is the amount of metric tons, the area. At the top, we have green peas. Interestingly, China is by way and above anybody else as the main producer of green peas. Dry peas produce a large tonnage as well, and for that, Canada is the main producer of dry peas. But then you might ask the difference, what's the difference between field peas and dry peas? And the main difference is the garden peas and field dry peas is that garden peas have a higher sugar content, and those are the ones we get as frozen peas. And field peas, which produce dry peas, have a much higher starch and low sugar content. And if you're ever a tourist in British pubs, you're offered something with mushy peas. Mushy peas are dried peas, rehydrated and dried green, and they are a British delicacy. Interestingly, most field production at field scale, whether for fresh peas or dried peas, uses leafless peas. These are peas with no leaves. They just have tendrils and stipules. And the reason these are used is they don't have to be staked. They hold themselves up in a vast mass. If you put normal peas in a field without so many tendrils, they just fall over, they rot, they can't be harvested. So this is pea production, and this is then combined. There is the production of green peas in Italy with the combine, and here are dry peas being combined in Canada. Now, the story of the importance of peas to biology, of course, really begins with Gregor Mendel. And there is a portrait of Gregor Mendel. holding his favorite plant, a pea. And one of the main genes that I worked on in this is the tall pea versus dwarf pea. And we are pretty sure that the lines that we worked on are the same as worked on by Mendel, because we had pretty good records of what was being grown at that time. Now, Mendel was a monk, an Augustinian monk in an abbey in what was then called Brunn in the Austro-Hungarian Empire, and it's now called Brno in the Czech Republic. So, graduate students, can you recognize who on that is Gregor Mendel? Anybody like to suggest? Stick your hand up and describe where he is. Okay.
He's in the back row, second from the right. He's holding a pea plant.
Very good. However, look at the jealousy of the surrounding monks. Now, I trust when you go to the dining hall, you get these looks of jealousy because you are working on plants, which makes you the most interesting graduate students around. However, we then have to ask the question, what is he holding? So there's an enlargement of that. What do you think he's holding? He's not holding a pea plant. Look at the flowers.
Fuchsia.
Fuchsia, thank you, Nina. He's holding a fuchsia. Just happened in that photograph, he was holding a fuchsia. And of course, Mendel is very famous for his original rules of genetics, and I'm sure you're all familiar with this from high school. But here are the modern Mendels. These are the current pea researchers. Well, actually, Ian Murfitt is retired. And these, I know everybody on this one, except I have never met Noel Ellis. These three are University of Tasmania, and there has been a close cooperation between Cornell and Tasmania. Ian Murfitt did a sabbatical up at Geneva in the 1970s. Jim Reed below did 2 sabbaticals at Cornell, and as a good friend of mine, I did 2 sabbaticals in his lab. Jim Weller is probably the most noted pea geneticist at the present time. I better say pea molecular biologist, because genetics has moved on. He does more of the sequencing and gene identification in peas than anybody else. Noel Ellis worked on the molecular biology of leaf formation. Christine Beveridge worked out the control of branching, and she's at the University of Queensland. And Jocelyn Osger in Alberta works on seed biogenesis. Here are all Mendel's genes. I don't have time to go over every one, but if anybody wants any of this data, it will be in 2 places. A, these slides are being saved, and secondly, Chris Ween's book on vegetable crop physiology comes out next spring, and it's all, whoopsie, and it's all in there. So here are Mendel's genes, or the traits. Here are the, and the different phenotypes. Here is what we know the genes are. Here is the change at the molecular level, but there are still 3 genes that Mendel worked on that we don't know really what they are. But many of the others we know, round versus wrinkle, tall versus dwarf, etc. Pea has a standard hypogeel germination, which you'd be quite familiar with. In the dark, the seedling stems grow much taller than in the light. Light causes a very pronounced decrease in elongation. And if they're growing normally in soil and they're 2 or 3 inches under, you get this shoot that comes up through the soil. The apical bud is hooked and was one of the classic bits of research involving ethylene, the apical hook. And when you put it in the light, the apical hook unfolds and you get the generation of the expanded leaves. Now here's some work by one of my graduate students, Fred Beringer. And what he was doing was tracking the growth rate of the stems second by second with a very sensitive system. And looking at the change in growth when you turn on the light. So up the side, we have the growth rate. On the bottom, we have hours after exposure, and the light came on at 0. Here's the result of exposing them to red light, blue light, or white fluorescent light. Here's where the light was turned on, and you get a drop in the growth rate within about a minute. A very precise, very rapid response. If that occurs in white light or blue light, if you have red light, the response takes at least an hour to an hour and a half before it shows any effect. And then following about 3 or 4 hours, the growth rate recovers to certainly a lower rate than the dark, but it does have some recovery after going with blue and white light almost down to 0 for a couple of hours. Now, what is interesting is what happens if you give the seedlings a pulse of light. So we tried that, and so here they got a pulse of 8-second fluorescent, and you get this immediate drop. But if you've only left the light on for 8 seconds, the growth then goes back to the dark rate, and then at an hour and a half drops again. So this is dissected out very nicely, the fact there are 2 different receptors here. Cryptochrome picks up the blue light and has a very rapid effect. Phytochrome picks up the red light, but doesn't produce any effect on growth till about an hour and a half. What we find is that the regulation of the growth rate depends on the level of gibberellin, particularly gibberellin number 1. And if you take a dark-grown pea and measure the gibberellin content, this is the gibberellin content. Here's the level in the dark. After 4 hours of light, you have to give it enough to see these changes measuring gibberellin. The level drops and it stays there. And then the level goes way up in the light. Now, I've already told you that the growth in the light is much less in the dark. So how can this be? Well, if this was just looking for the sensitivity to gibberellin. Dwarf peas were taken and treated with an extract, treated with gibberellin to see how they would respond. And dark-grown seedlings respond a large amount. However, after they have been in the light for just 1 day, they are much less responsive to gibberellin, and in constant light, they are also less responsive. So what we are having when the light comes on, first of all, there is a rapid drop in the responsiveness to gibberellin for the gibberellin signal transduction chain. Then the level of gibberellin actually falls. Then the sensitivity stays low as the level of gibberellin goes back up. And that reflects very nicely that instant drop, and then a few hours later, the growth rate comes up somewhat. Peas have been used to look at chloroplast changes as the plants come into the light. And this is just a few pictures. So here's the results of some electron micrographs looking at the chloroplasts in these peas. And this is in darkness. And here you see the changes 8 hours, three days later, and you see the membrane starting to form. And then in the 4th day, you see the membranes looking much more like a typical chloroplast. And a thorough analysis was done. This is enlarged, just enlargements of each one. And so what we find is that some of the photosynthesis components are actually present in the seed or the seedling, the dark-grown seedling. Others only appear after the light has been turned on or the plants move into the light. Now, I spent quite a bit of time in my career looking at pea internode length. And here we have again Mendel's peas, the tall pea versus the dwarf pea. If we look at the difference in growth rate, I should point out the difference is not the number of nodes, it's the length of each internode. So this now looks at the internode length in the y-axis and internode number on the lower axis. As you can see, the internodes do, as the plant grows, they do get slightly longer, but they peak out at a very big difference. This is millimeters, so about this is about 4 centimeters, 3 to 4 centimeters here for the dwarf, and about 15 centimeters for the tall. Big difference. So what is the reason for this? Well, what my colleagues and I discovered way back, we're talking about the 1980s, is that the crucial difference is that dwarf plants have a very low content of gibberellin number 1. Tall plants have a high content of gibberellin number 1. And the crucial difference is the transfer from the precursor of gibberellin number 1 to gibberellin number 1. And the precursor is gibberellin number 20, GA20. And the difference between GA20 and GA1 is simply one hydroxyl group. GA20, it's not, GA20 here is inactive in growth. It doesn't have any effect on growth. GA1 is the growth active gibberellin. And there is an enzyme that puts on this hydroxyl group, and it's called GA3 beta hydroxylase, or more recently, they've just renamed it GA3 oxidase. And it is called that because as the numbering of the carbons in this molecule, this is carbon number 3. So it's just the addition of one hydroxyl makes all the difference between a tall and a dwarf. They both have GA1. A dwarf has a low level, a tall has a high level. So my colleagues and I then were able to isolate the gene involved. This is 20-something years ago, 22 years ago. So a lot of this is history. We were able to isolate the gene and show that there was just one difference in the gene, a single base change. If you between a G and an A, that this led to a change in one amino acid, and this single amino acid change made all the difference as to whether the enzyme gibberellin 3-beta hydroxylase had high activity or very low activity. And to show this, we then put the gene into the bacterium E. coli. E. coli doesn't normally make gibberellin, but when you put the gene in, it will take the GA20 and it will convert it to GA1. So we did this with the gene from the tall plant and the gene from the dwarf plant, and then looked at how much metabolism, if you then fed GA20 to the bacterium, we asked how much GA1 would it make, and here is the result. This is high performance liquid chromatography. This is the amount of radioactivity. This is the position where GA20 comes off. GA1 comes off much earlier off the column. And so here is the conversion of GA20 to GA1 in tall plants, and here is the conversion of GA20 to GA1 in dwarf plants, neatly showing that indeed we were correct, that the enzyme, the mutant enzyme in the dwarf plant is much less active in this transition. So here is the pathway for gibberellin biosynthesis. And the last step is down here, GA20 to GA1. The gene is was designated LE for length, and there's the enzyme, and that's what Mendel was working on some 160 years ago, but he didn't know what he was working on at that level. But it makes a very nice story. Now, there turned out to be, there are a whole lot of length mutants. And an artist grew these from peas I grew in the greenhouses here. This is an extreme dwarf, which is called Nana. This is Mendel's dwarf. This is Mendel's tall. And the last one is a super tall. And this super tall turns out to have a mutation in the gibberellin signal transduction chain. Now, with regard to gibberellin and auxin and many other controls in development, the null with no effect in this case is tall. When you have, there's a signal transduction component which we call DELA, and DELA simply is a protein, it's a transcription factor, and DELA simply stands for the sequence of amino acids at one end of the protein. In the presence of DELA, the tall becomes short. You give it gibberellin, it negates DELA, and it becomes tall. This one has a mutation in the signal transduction chain component. So regardless of the level of gibberellin, it is a tall. And we now know all the controls in this system. So here are some of the gene symbols. And these, the lighter green, are the names of all the enzymatic steps. You can also get a very tall by blocking the degradation of a GA1 or its precursor. And if you block the degradation, you get a higher amount of GA1 simply because it has nowhere else to go. Then you get an extra tall. Here are a whole lot of other height mutants. This is the wild type. This is super tall. This is a tall. This is a super tall. And this one involves phytochrome signaling. The next one is a phytochrome mutant itself. The next one, this is the DELA signal transduction mutant. This one is another wild type on a different background. And here is this plant where the breakdown of GA1 has been inhibited. Here are some more mutants. These all come from Jim Weller, who has great fun getting all these mutants. This one, this by the way, is in the light. This is in the dark. This one, EIN2, is an ethylene receptor mutant. There it is in the light and the dark. This one is involved in ubiquitin, which is the degradation of various signal transduction components. And the last one is a phytochrome mutant. If we turn to the roots, we go look under the soil. It has a taproot, but a very branching root system. And most of the mutants that we worked on here, a lot of work was done here by Tom Silver, who teaches the plant physiology labs and the botany labs here now. Only if you had the very deficient gibberellin, very deficient mutant NANA, did you get smaller roots. Every other mutant of the top of the shoot, the roots look exactly the same as this. One of the classic things that was discovered about auxin many years ago is if you take a plant like a pea, a corline plant, and you cut the apical bud off, you then get branching. And this has been was attributed for many years to the fact that you'd removed the source of auxin. It turns out that auxin is part of the story, but not the main part. When you cut the apical bud off, actually the sucrose that has been made goes now to the lateral buds. And sucrose has a big effect on growth. So sucrose is not just sort of an energy source, it has some growth promoting effects of its own. This was all worked out by Christine Beveridge and co-workers. But auxin comes from the top, and it was said that auxin inhibited the lateral buds. Turns out there is some inhibition of the lateral buds, but it's not by auxin itself. The timing of the changes in auxin and the lateral buds didn't work out. Auxin goes to the roots, and there it stimulates the production of another group of plant hormones called strigolactones. Strigolactones, the name is derived from the parasitic plant striga, and these were known to promote the germination of striga. And there's a family of these hormones. It turns out the auxin goes there, causes the production of strigolactones, which inhibit branching. When the auxin supply is cut off, the level of strigolactones goes down, and you get the branches formed. This was a story that stumped plant physiologists, biochemists, molecular biologists for many, many years, but it was finally solved about 10 years ago. Next thing is peas have many different leaves. And here are the main types. And these are all genetically controlled. I am not going to discuss the individual genes. If anybody's interested, we can go back to it because it would take too long. But it is interesting that there's not just one type of leaf. You can have, this is the standard pea leaf. You can have more leaflets and no stipules. You can get rid of all the leaflets, and no tendrils. You can get rid of all the leaflets and the leaflets become tendrils. You can have multi-branches and tiny little leaves on the end. So you can have all sorts of combinations. There are also genes that turn the stipules on or off and control stipule size. And here's this semi-leafless pea that is the main pea of commerce once you get out of your home garden. They have put in the gene, or actually mutated the leaf gene to make all these stipule tendrils, but the stipules have been retained. Other peas were produced with no stipules. Because on the idea, well, if you get rid of them here, maybe getting rid of these, there's some photosynthesis by the tendrils. The idea was, well, let's get rid of all the stipules as well, and then the light will penetrate the crop. Well, it turned out that doesn't work. The yields plummet when you get rid of the stipules. So these tendrils hold the whole thing up, and now the main photosynthesis is being done by the stipules. Here's what it looks like under the scanning electron microscope. These are from Noel Ellis and his lab. It's not quite the same magnification, but this is sort of a wild type, just as an example. And here's one where the leaflets are being turned into tendrils. So you can see the young tendrils sort of coming up at the end of the stem. One thing that peas do that Arabidopsis does not, they nodulate and they do nitrogen fixation. And here all the pea roots covered with nodules. Now, when I was a teenager, I was into gardening. And one of the plants I grew was sweet peas. And I was transplanting them, and I saw these white blobs on the roots, and I was very distressed. And so I spent a lot of time pulling them off before I planted them. I only later learned what these were, that they were nodules. We also know a lot about the nitrogen fixation process and its control. It turns out that the control of infection and nodule development is very much under the control of hormones. And this diagram comes from a paper I wrote last year. But notable amongst this is gibberellin actually inhibits infection of the root hair, but promotes the development of the actual nodule. Okay, if anybody has any questions as we go, feel free. I'll do my best to answer them. So we move on to flowering. Now, there are huge numbers of genes that affect flowering. This is a wild type with no mutations. This is a mutation in the SN gene, and you can see the difference. This is flowering much earlier. Turns out the peas are photoperiodic, the wild type pea is photoperiodically sensitive. And if you grow these, they will not flower until midsummer. They like long days. But peas are also a cold season crop. They grow much better around here if you grow them in the spring, in the cold, rather than in the middle of the summer. And it turns out that virtually all our pea varieties are SN, because this was, as peas, they thought to originate in Asia. As they moved into Northern Europe, the early flowering was selected for so that they produced a crop early on before midsummer. And that is one of the proposals. This mutation led to the adoption of peas much more widely than would have been the case. So I won't go through all these mutations, but here's some interesting ones that are related to what you may know about the control of flowering. First of all, late one, here it is in long days here. Late one is, I'm wondering if I made a mistake there. They're both labeled as long days. I should check that. Late one is a clock-related gene involved in circadian rhythms. Gigas here is equivalent to the gene FT in Arabidopsis, and FT is the gene which controls the production of florigen, or the flowering hormone. VEG2 here is equivalent to the gene FD in Arabidopsis, and FD is one of the genes that's turned on by florigen when it arrives at the stem apex.
Do all these mutations have corresponding effects on the root system or nodulation?
Can't tell you. No idea, Ken. What we do know is the roots do stop growing when flowering starts. One of the things I had growing many, many years ago at Goodman, I built a glass-sided, actually plexiglass-sided trough sloping inwards and planted the plants at the top with the idea I could follow the root growth. I made it, got a 4x8 sheet of plexiglass, it was 4 feet. Planted the peas, filled it with Cornell mix, and the peas, when they were about that high, 10 centimeters high, the roots reached 4 feet. So the depth of the roots is just amazing, and it occurs very rapidly. Now, Cornell mix, I presume a lot of you are familiar with, it's a very light, fluffy mix. So it probably doesn't exactly match what would happen in a nice stony soil around here, but it blew my mind. I ended up getting long drain pipes and trying them to 12 feet to see what was happening. But the interest, again, it's very early on the roots grow. And then when flowering starts, root growth virtually stops. So it's all during the early vegetative part. But what these mutations do, I don't know. This is Jim Weller's photograph. Maybe I'll have to ask Jim to measure. He's going to tell me. I'm too busy with other things. I can just see it coming. Here's a range of, there's a lot of different branching patterns as peas flower. And here's the basic idea. The corline stem has an apex which keeps growing. We call that I1, a primary meristem. And off at the axillary buds, another branch starts, and that has a meristem which we're calling I2. And these are both controlled by various genes that produce signals, and these all have to interact together at the correct time. And if you mutate any of the genes, these genes, you get a very different range of inflorescences. So there's for example, here's a gene PIM, and this has proliferating I2s. The I2s keep branching off here. In DET, the primary inflorescence becomes an I2 instead of an I1. So it has some flowers off here and then stops. And this one, veg, all the I2s have become I1s, and nobody told them they had to produce flowers. So here's a list of the different flowering loci in peas. In the interest of time, I think I will skip that. And to tell you the story of plant senescence, which I worked on for many years. Here's Jerry Marks at Geneva in the 1970s. There were two centers of Pea Genetics, Tasmania and Cornell at the time. And we were interested in plant senescence, and he alerted me to the fact that he had some peas that senesced in summer but not in winter, and wanted to know what was going on. And we found indeed they were senescing in long days but not in short days. And the name of this variety was G2. Here's what you see. The apex keeps growing in short days but not in long days. And during a sabbatical here and his subsequent work, Ian Murfitt, working with Jerry Marks, worked out that there were two particular genes that were in a dominant form that gave this delay in senescence in short days. It was nothing to do with the number of fruit, because we got short days produce just as many fruit, though if you do remove all the fruit, it will keep growing ad infinitum. So it does need the presence of fruit. There's one in short days, huge numbers of fruit, rapidly growing, no problem. So we wondered, well, could fruit be sending a senescence factor? So we labeled peas with CO2, radioactive CO2, and then asked whether radioactive is getting into some compounds that go to the apical bud and kill it. And here's where the radioactivity went. Most of it went to the leaves, and it turns out it goes to the leaves by reverse xylem flow. And that also happens in apple and similar crops. The sucrose solution gets into the apple, and the water leaves by the xylem, the opposite direction to normally expected, but very little in the apical bud. And when we analyzed the apical bud, the radioactivity, it was in sucrose and malic acid. No evidence for a senescence factor. So we then went and looked at the morphology, and actually what you see is a difference in speed of flower and fruit development. In long days, the flowers and fruits develop much more rapidly. So they are getting a signal that they are not getting in short days. And so here the apex keeps growing. Here it starts producing mainly flowers and fruits. And what was happening is that this is the amount going to the reproductive organs, the amount of photosynthate. The photosynthate going to reproduction is much greater in long days than in short days. I will skip that, and I'll skip that. So we then looked at the hormonal control, and we find that this is the amount of gibberellin going to the shoots as distinct from the reproductive tissue. And this is GA1. The level of GA1 in the apical buds in short days was much greater than in short days. So our conclusion to many years of research actually, was that senescence of the plant is determined by hormonal changes that direct the resources of the plant towards seed production at the expense of continued vegetative growth. And these are two handsome young guys working on peas some over 30 years ago in the greenhouses at Goodman. So I know it's time, but I got a couple of little neat things that I thought you might be interested in. So what I'm going to call pea fakery. Peas are also a favorite topic for people I'll call pseudoscientists. So the first question was asked by Mythbusters. Anybody ever see that? Used to be a very popular program on TV where they investigated claims. I haven't seen it recently, but Mythbusters wanted to know whether plants preferred rock or classical music. So they put the peas in the greenhouse, and in one greenhouse they played rock music, in the other one classical music. Then they took the plants out. Both plants looked absolutely miserable. But it wasn't the music. They had them in greenhouses in the Central Valley of California in the summer, and they were cooking them. So when you look at any scientific report, look at what the conditions are, because some of the results may have nothing to do with what they claim to be moving. So our answer is they don't care. This one is in the last year. There is a woman researcher in Australia who actually has a faculty position, much to my amazement. And she claims that plants have sensitivities. And she claims she got a grant. An oak tree told her she was going to get her grant. And one of her things is she said plants can hear, and they can hear water in pipes. And she's been on NPR. She got a full page write-up 2 weeks ago in the New York Times. And here's her experiment. She puts a little tube of water flowing under here and none under here, plants a pea at the top and says, oh, look, the roots grow into the side with the water pipe. Isn't they hear the water? Okay, graduate students, look at that picture. What's wrong with it? Well, to start with, it's one pea, and pea seeds are not symmetrical. She planted the seed centrally, but the root comes out on one side. Oh, look, it went down that side. Oh, that's going to get me on NPR, right up in the New York Times. This is what I call junk science. Be aware of junk science. So, sorry, I did go 2-3 minutes over, Don, but you got through the entire thing of peas. Here's my colleagues over the years that worked on peas, and for which I thank all of them, and it couldn't have been done without them. So I hope at least you know a little bit more about peas than you did coming in. And they're such exciting plants. Okay, thank you.
This has been a production of Cornell University, on the web at cornell.edu.